The overall goal of this procedure is to harvest interneuron precursors from distinct brain regions in postnatal mouse pups and transplant these cells into age-matched recipients to assess how environmental changes influence interneuron fate and maturation.
This method can help answer key questions in the field of developmental neuroscience about how intrinsic genetic programs and environmental signals interact to sculpt the fate of neural cells. The main advantage of this technique is that alterations in the fate and maturation of interneuron precursors can be analyzed after their adoptive transfer into a new brain environment.
Visual demonstration of the cell harvesting and grafting steps is critical because small errors in the technique can lead to inefficient and unsuccessful transplant experiments.
To begin, place one razor blade through the anterior frontal lobe of a postnatal day 1 pup brain, and additional razor blades into the slots just posterior to the first razor blade to generate two 0.5 mm slices. Transfer the striatal slices into a petri dish with bubbled sucrose artificial cerebrospinal fluid, or SACSF, and place the remaining brain tissue into a petri dish with SACSF on ice. Place the slices under a dissecting microscope and use forceps to pinch the striatum from both hemispheres, placing the striatal chunks in a 50 milliliter tube of SACSF on ice as they are harvested. If the brain was harvested from a fluorescent reporter mouse, use fluorescence microscopy to distinguish the striatal